Uh, Thank you for the introduction. Thank you, Mastek, for the invitation. I'm Alice. I'm a postdoctoral researcher in Negri Institute in Milan. And as Enrico mentioned yesterday, we are part of a mass spec center research focused on health and environment. We have different platforms, but as you can imagine, today I will talk about our imaging mass spec applications. And no many spectra in my presentation, but I will tell you something about two different stories. The first one will focus on my PhD project. I spent 4 years studying Huntington's disease. And the second one is a part of my postdoctoral project focused on the leiomyosarcoma. In the first case, we will show the study of the spatial distribution of cholesterol in brain from R6 to mice, that is a mouse model of Huntington's disease. And in the second study, I will show you how we can use the MALDI mass spec application to study the spatial distribution of docetaxel in tumor. But let's move step by step. Brief introduction about Huntington's disease. Huntington's disease is a dominant neurodegenerative disease described for the first time in 1872. It is caused by a mutation by Huntington gene that causes the translation in a Huntington protein that is mutated. Huntington disease is characterized by neurological and peripheral symptoms such as the motor symptoms, the cognitive deterioration, and psychiatric problems. But we can have also other symptoms such as weight loss or atrophy of skeletal muscle. But as I mentioned, our focus is cholesterol, and cholesterol is essential for the maintenance of brain homeostasis. In fact, we know that the brain is the richest organ in cholesterol. In fact, the 80% is localized in myelinated sheets, and the 20% is the structural component of astrocytes. Every day our brain produces new cholesterol that is essential for maintaining the synaptic vesicles. So we know from literature that in the last 20 years several papers were published on the Huntington disease and the correlation of neurodegeneration and cholesterol. That is, Huntington disease is characterized by an impairment in cholesterol homeostasis. So I started the study of cholesterol metabolism in brain from R6 to mice using a classical ASMS method. And as you can see, we have a classical method with bile acids in the first part of the chromatographic run, the separation of the oxysterols, and the elution of the cholesterol precursor. Briefly, we found the reduction in brain from R6 to mice in desmosterol and in 24S-hydroxycholesterol. That is the main catabolite of cholesterol brain synthesis. But when we use the ASMS method to study the total content of cholesterol in brain, we did not see any difference between wild type and R6 to mice. This was quite strange. So we decided to have a new approach to study the cholesterol alteration in brain from R6 to mice. And for this reason, we developed an imaging MS method for the study of the spatial distribution of the free fraction of cholesterol in brain slices from R6 to mice and wild-type mice as control group. To obtain these results, we set up this approach starting from a published paper from Swansea University, where I spent 3 months. And we use the SunCollect sprayer to perform a 2-step derivatization. We sprayed, using the SunCollect spray, the cholesterol oxidase, and then we sprayed the JRT reagent, that is a derivatizer, that allows us to increase the signal of cholesterol in AP-MALDI source. As cholesterol had a poor ionization in AC source, also in AP-MALDI the signal was quite low. But after this reaction, so the first reaction of cholesterol with the reaction after the incubation with cholesterol oxidase and the reaction of our product with the GRT reagent, we obtain a molecule with a nice signal in our instrument. So this is the result. On the left side we have the wild type slice brain slice, And on the right side, we have the slice from R6 to mice. We can appreciate a reduction in free fraction of cholesterol, but to better understand this difference, we decided to have a quantitative approach. So starting from a previously published paper from our lab, we perform a quantitative imaging mass spec approach. We use a calibration curve spotting directly on target increasing concentration of cholesterol, and we sprayed an internal standard, in this case cholesterol D6, all over the target of IP-MALDI. And the results allow us to appreciate the differences between wild type and R6 to mice. And our results suggested that although the total content of cholesterol in brain is maintained at the steady state, as we found using the ASMS method, the available fraction of cholesterol is actually affected by the reduction of cholesterol synthesis in brain. So in conclusion, for this fourth part, my aim is to show how the results obtained with the integration of two different mass spectrometric approaches allow to have a deep understanding of the metabolic alteration related of a pathological state. In this case, the use of IP-MALDI imaging approach allows us to appreciate the difference between wild type and R6 to mice in terms of cholesterol brain content. In this case, I would like to thank all the collaborators of this huge project on Huntington disease, starting from my colleague of Mario Negri, Università degli Studi di Milano, Istituto Carlo Bessa, and Swansea University for the help in setting up the imaging mass spec method. But as I mentioned, our second example is related to leiomyosarcoma study. Also in this case, a brief introduction. Leiomyosarcoma is a type of rare cancer that grows in smooth muscles, and it is very aggressive, and it is found most often in abdomen. It can occur at every age, but in most of cases we found it starting from 50 years. And the symptoms are common, and they include abdominal pain and unusual bleeding. Regarding the treatment of this kind of tumors, surgery is the preferred treatment, but actually we have also the chemotherapy for provide survival benefits in case of resistance. And also for the metastatic disease, treatments can include the drug treatment using also a combination of drugs. And our focus is related to docetaxel treatment, and in particular we want to evaluate if the pre-treatment with eribulin, another anti-tumor drug, can improve the distribution and penetration in tumor tissue of docetaxel. So this was our experimental design. At day 0 we treated animal with eribulin, and we did the same at day 7, and after 5 days we treated animals with docetaxel. Also in this case we use a quantitative imaging approach, so we use the sun collect sprayer to spray the internal standard and the matrix as well. And this was the results of our calibration, so we use a control tissue without docetaxel concentration in it, and we spot on it different concentration of docetaxel. We spray the internal standard, that is paclitaxel, a similar drug, all over the tissue, and we plot our calibration curve. These are the results. On the upper side of the slide we have the distribution of docetaxel in tumor tissue, and in the lower panel we have the docetaxel distribution in tumor tissue after the treatment with eribulin. And we have a significant difference in concentration and penetration of docetaxel after the pre-treatment with eribulin. We deeply investigated the reason of this improvement in docetaxel penetration and distribution, and we found that eribulin pretreatment can improve the vascularization in tumor. So the consequence is that we have a better tissue for anti-tumor drug distribution. So In the first part, in the upper part, we have the results from docetaxel treatment, and in the lower part, we have the results with the pre-treatment with eribulin. So in conclusion, in this case, the eribulin pre-treatment improved the penetration and distribution of docetaxel, and we can appreciate these results using the imaging mass spec approaches. Future study, we will focus on definition of a treatment scheme in terms of doses, and also we want to investigate the metabolites in tumor to understand if any altered metabolic pathway could help this kind of distribution in tumor. So for this part, I would like to thank all my colleagues as well from my lab, Istituto Europeo di Oncologia e Humanitas Research Hospital. So in conclusion, mass spec, starting from our application on mass spec imaging, we can say that in the first case, I show you how the use of imaging mass spec, how to understand the alteration of a metabolic state in brain, in particular for the cholesterol, the use of imaging approach allow to see a difference that we cannot see using CMS. In the second case, the use of imaging mass spec for pharmacokinetic studies, in this case, allow to see the distribution of drug, not only the concentration in tumors, but the distribution and penetration of drug at the target site. So I would like to thank my mentors, Enrico and Renzo, and all my colleagues for MassTech Lab, MassTech for the kind invitation today, and all of you for your attention. Thank you.